doing and probably with some future perspectives. With these words, I hand over the microphone to my colleague Dr. Vishwanathan, and he will proceed.
Good morning. It's a pleasure to organize this session. We have some outstanding speakers, and since we're really short of time, I think we should get started. Dr. Sharma will introduce the first speaker, please.
I am the first.
Dr. Sharma is the first speaker. How about that? So Dr. Sharma is from Uppsala University, Sweden, and he requested that we don't give long introductions to any of the speakers. So here he is on his part.
My first name? Okay, you are using the same computer. So, again, this is a pleasure and honor to be here. I will briefly tell you our research program that is conducted in Uppsala University together with different organizations, notably US Air Force Research Laboratory, European Office of Aerospace Research and Development, National Center of Toxicological Research, my university, Uppsala University, more than 600 years old, and with Swedish Strategic Research Organization. Our idea is that, my dear friends and colleagues, that we must understand nanoparticles with their unique features, everywhere they are. And before I tell anything, I must admit and tell you that whatever I am telling, this is not the official version of US Air Force Research Laboratory or US government or Swedish government or any organization that are involved with this. So what we believe that I am interested only in brain functions, and we have nanoparticles everywhere in the environment by motor vehicle exhaust and many other things, pesticides. So what will happen that if we are using or inhaling these nanoparticles, and if we get some disease, for example, then it will produce adverse mental situations. In these circumstances, this blood-brain barrier we all have is playing a very important role. And as you can see, that our central nervous system is composed not only with nerve cells, but lots of glial cells. And this is the capillary and brain interaction where the blood-brain barrier resides. I will ask the question to you that do you understand, for example, if our soldiers are posted in somewhere in desert environment, or even in Afghanistan, they are breathing several nanoparticles from the environment. If they get traumatic injuries, do you think it could be same? No, it could not be same. Again, in many parts of the world, we have pollutions in the environment. And do you think that it will affect also normal healthy persons and persons having hypertension or diabetes equally? The answer is probably no. So we are working on this thing. We believe that any insult on the central nervous system caused by nanoparticles will open the barrier, produces many changes in the brain leading to cell death. It means early neurodegeneration. We have produced this book for making awareness, Nanoneuroscience and Nanoneuropharmacology in 2009 by Elsevier, telling that this is indeed a problem. We also have a journal called Journal of Nanoneuroscience published by American Scientific Publishers, recently introduced, and these are the editorial board members. You have heard Dr. Kalam yesterday, and he also emphasized nanoscience and nanoparticles between space and Earth, and we are working in this area with Sweden, USA and India consortium. These nanoscience work is also supported by politicians here in India. This is Sonia Gandhi, and also equally Anders Schally, who won the Nobel Prize in 1977 for physiology and medicine. So we believe that we cannot ignore the problems of nanoscience or neuroscience. This is the Nobel laureate in chemistry. He also feels that this is the area one should study. So we have just compiled this review. It's multidisciplinary John Wiley review, where we have emphasized that effect of nanoparticles may not be same in healthy individuals or in persons having diabetes or hypertension. That is very much prevalent in our country, for example in India. I am basically a neuropathologist and learned my techniques from Professor Ingve Olsen much earlier, so I have interest in pathology. And what we believe that at this time we cannot ignore nanoparticles in neurotoxicity and even nanodrug delivery. Think like this situation. Our soldiers are exposed to silica dust, and if they get any kind of injury, probably the consequences will be much different. Most of the soldiers, they have to be like this for a long time. So this is also one kind of immobilization stress. So this is a combination of various things. I will show you some results from my laboratory on hypertension nanoparticles, and these are the equipment where we have cognitive and sensory functions evaluated. You can see here, due to lack of time I cannot state more, but you can see here that hypertensive animals showed reduction in performance, and when they combine with nanoparticles and stress, these functions are further deteriorated. The same thing also happening on inclined plane angle. The combination of stress, nanoparticles, and also here at 21 degree and 33 degree. If the environmental temperature is higher and they are consumed with silica dust and given hypertensive, their functions is much more deteriorated here. And so therefore, silica neurotoxicity is higher at 33 degrees Celsius. This is an example of nerve cell damage, not going in that detail, but you can see a combination of hypertension, silica, and immobilization stress at 33 degrees Celsius. Neuronal damage is highest in many regions. This is the example here. You can see silica and hypertension 21 degrees Celsius, silica and hypertension 33 degrees Celsius as compared to the silica alone. So if nanoparticles are exposed at high ambient temperature, it is much more stressed than normal situations. Now I will show you some example of diabetes, how it can affect. So this is an example of diabetes in saline. These are the leakage of serum protein, making opening of the barrier. When we added silica, then this is leakage of barrier is much more higher, and a combination of diabetes and silica, it is much extensive. So nanoparticle and diabetes combination opens the barrier, making neurons more vulnerable. This is an example. This is the combination of diabetes and silica. You can see there is vacuolation as compared to the normal situation. At electron microscopy, you also see that these central nervous system myelin damage is much more prevalent here as compared to the other situations. So the take-home point is that if persons having diabetes are exposed to nanoparticles, more central nervous system damage will occur. Now, my wife has done some experiments on age and size related experiments, and this came as a hot news in neuroscience this year. So, going briefly, we have different kind of size range of nanoparticles and different kinds of age. Not going in methodology, I will tell you one thing, that the small size of nanoparticles, they are doing most harm, and this is much more prevalent in the young age and the older age, 9 to 10 weeks compared to 30 to 35 weeks. And this is particularly quite common for even copper, but aluminum is, though the pattern is same, but aluminum is least neurotoxic. Then we studied cell changes, and you can see here that young, adult and old, copper 50 to 60 nanometer, nitric oxide synthase of regulation was maximum in these two situations. Astrocytic activation was also much higher in these two situations, and neuronal damages. So we can see that the smaller the size of nanoparticles can produce higher the damage, and this is especially prevalent in young age and older age group. Then we tested our hypothesis at electron microscopy, and you can see here this is myelin basic protein. Young and old were highly susceptible to silver 50 to 60 nanometer, and this can also be seen at electron microscopy. These are the myelin vesiculation. This is damage. This is much more damage, but these adult group is also damaged, but much less. Now in last 2-3 minutes, I will tell you the latest technology that we are using, and in this situation, you have heard that Parkinson's disease, they are using deep brain stimulation, and in this situation, If you use normal electrodes, there is always this accumulation of glial cells and spot that prevents the stimulation. So therefore, NASA has developed this nano knife, and this is not a fiction. They are using it, and also the microelectrodes. This one of the microelectrodes are carbon nanotubes, and they combine both nanowire and nanotube. And this is an example that cells can be stimulated with multi-electrodes. I have less time. I will provide some nano drug delivery results to you. And in this situation, you can use any substance for nano drug delivery. This is carbon nanofiber. And you can use particularly any drug. We have some experiments with cerebrolysin. And this is, I'm not going in detail. Dr. Ryan Tian is the person who in Fayetteville has the patent of nanowire. And this is Dr. Young, who is the leader of nanowiring. And stem cells can even also be nanowired. I skip this. We are using titanium nanowire for drug delivery. And these nano drug delivery can also produce the cell damage. So making this point that if we use this nano drug delivery, we can have this less cell damage. And this is also supported by various administrators in the United States. I will skip this, thank you sir. The traumatic injury is also higher after silica. And this is the Arkansas governor supporting us. I skip this. And we have different supports. We are also working with Chinese government and military. And these are the different organizations for support. Thank you very much for your kind attention.
Can I ask?
Yeah.
I am Dr. P.L. Nayak, Director of Nanoscience Diagnostics. Well, I have a question. Wherever I go, I ask, what is the future of nanomedicine? Whether we have reached the destination or we are still in the journey? Because we have talked a lot of things about nanomedicine.
Nanomedicine has a distinct future.
Yeah.
What we are doing together with US FDA and US Air Force Research Laboratory to understand the toxicological aspect.
Yeah.
Because of lack of time, I did not show you the results that we are using nano drug delivery of two beautiful compounds, and we can, using right type of nanoparticles, if we have this drug delivery of right kind of medicine, then one can enhance the neuroprotective effects of those drugs. So in those circumstances, they are having good future. But before it can come for normal routine and practice, we must understand its toxicological aspect.
What I feel as a worker in this field, the only thing which you have succeeded is the drug delivery, controlled drug delivery.
Yeah.
But we have got very good results also throughout the world. And I think that is the only field which now we should give more emphasis, because in India, such a poor country, and drug delivery is. What is your feeling about this?
Yes, nano drug delivery is a very important tool for the treatment. But as I said you earlier, we cannot ignore the toxicity caused by nanoparticles. Before we have done this, it is very difficult to implement in human situations.
So do you think that all the nanoparticles are toxic?
This is very difficult to say. As I told you that it depends on size, it depends on age, and depends on so many factors. But as pharmacological books say, everything is toxic depending on its dose.
Thank you.